So we can get even more complicated. So that's just Brownian motion. Now this thing is what's called a molecular motor. These are used in cells. So in fact, there's a specific example in a nerve cell. It turns out that the places where proteins are produced tend to be far from places where proteins are used. So therefore, molecular motors are used to convey those proteins from the places where they're produced to where they're used. So this is what's called a microtubule, and this is the actual moving element. This is called kinesin. Kinesin. I'm actually not sure how it's pronounced, but I say kinesin. The motion looks like this. So here's your microtubule, and basically what will happen is the legs of the kinesin will flip back and forth like this. And so it'll basically just walk down the tube. You can do this two ways. If you look at it this way, what you can do is you can attach a cargo up here, and the kinesin will march down the microtubule, carrying the cargo along with it. On the other hand, the way in which I and my collaborators like to think about it is this. So we anchor the kinesin to some substrate, and the microtubule propagates along the top. This is more like so the other way was kind of like a railroad. This is kind of like a conveyor belt. So basically you have this two-dimensional surface that's lined with these guys. You drop a microtubule onto it, and away it goes.
It's like a Turing machine.
Kind of, yeah, exactly. So that, in one dimension, that's exactly right. And here's what that looks like. So here's a simulated kinesin walk. So now it's going much slower, but that's because my length base is much longer. This is now 20 by 3. And we can see that it's walking in an extremely regular way, and that's important. So the Brownian motion, it was just scooting all over the place. There was absolutely no regularity to its motion. Here, I mean, it's not going in a straight line, but it's certainly marching very regularly. So its motion is quite regular, and this has very nice properties that we're going to see more of later. So the idea with this one is that this is the lab on chip example. So you line an etched channel in glass with kinesin, drop some microtubules into it, and these would propagate.
So there's like, you're not shown some kind of lattice underneath here of these molecules that it's guiding itself with? Is that what's...
So how it works is that these things have a polarity, so I didn't show that, but there's a certain polarity, and they will either go this way or this way.
Yeah, but you're not showing the microtube there. I mean, that green
That's
and blue
correct.
surface is, they're climbing on something like that, right?
That's correct. But yeah, so right. So these guys are these are the motors anchored down the bottom.
Yeah.
What they'll do is they'll march in place on the microtubule and force that to them. So how this would go in an environment. What I'm doing here is I'm sort of showing this is a top-down view, if you like. So we're looking down on a channel lined with kinesin, and this length here, if you like, is the microtubule that we're sort of seeing propagate along from the top.
So in reality, which one moves? So the kinesin moves? Or?
Which one moves?
So I'm a bit confused. So I thought the microtube is fixed. It's the other thing that moves, right?
Right. So so if you're doing it this way, if you attach a cargo here, then this is like a rail, and this is like a train. So this is like in that case the kinesin moves. If you do it this way, these are the kinesins. They're all fixed to a substrate, and instead it's like a treadmill kind of thing. The microtubule is being forced along by these motors that are all stuck in place. So
maglev train.
Pardon me?
Maglev train, maybe.
Exactly.
But what
exactly.
happens in the cell? So that's what I was
Right. So in the cell, it's this way. In the cell, so in nature, it's this way.
Okay, yeah, and then you can model it whichever way.
Exactly,
Yes,
exactly. So this is engineered. This is engineered. I don't know if this actually occurs in nature, but what you can do is you can send a microtubule and you can actually attach a cargo to it.
And the reason it's not going in a straight line is because there's a two-dimensional array of these mechanisms.
Exactly, so it's not, I think that was the question earlier, it's not, once they have an initial direction, they tend to move in roughly that direction. But they're also affected, because they are small, they're much more massive than the molecule that was propagating before, but because they are small, they're affected by Brownian motions. They'll be knocked slightly off course, and that's why you have these sort of looping trajectories. So again, there is uncertainty in this motion, and we choose to use that as to model the noise. Now, there's much less uncertainty, much less variance in the arrival times here as there were in Brownian motion.